Okay, this is going to be our session where we review the basics of nerve physiology. As we all know, of all the tissues, there are only very few that are actually excitable, and that would be your nerve and your muscle. We have to start with the nerve because it is what commands the muscles to contract in the first place. And it is very important for us to review how the nerve membrane becomes excited or depolarizes, because that is how impulses are transmitted. And different diseases or conditions may affect certain excitability, and different conditions may make a nerve excitable or the opposite of that. Now, I'd like you um, to think about your central nervous system. Uh, it is not a simple nerve firing and basically commanding a muscle to contract. If you would look at the central nervous system, you would see a particular nerve, but you would also see other nerves surrounding it, and they are actually communicating with one another. One nerve might be stimulating the other nerve might be inhibiting this, and so on and so forth. It is a jumble of different neurons um, interacting with one another, and that what basically forms our our ultimately our personality. Now, just to give you an idea, last time we talked about the value of being able to draw. Just the fact or just the act of drawing while learning something is actually beneficial. And let me explain that. Wait, let me draw a bigger brain. Let's see, right now you are drawing with me while attempting to learn this particular lesson. Now, let's go back to your cerebral hemisphere. You still remember your important fissures, right? Right? You have your lateral, and then you have your central. Okay? So now you have your frontal lobe here, parietal lobe here, temporal, and then occipital. Now, this part here is your vision. In your frontal lobe, just in front of your central sulcus, is what you call your precentral gyrus. Your precentral gyrus is most important because it is your primary motor cortex. All the motor functions comes from nerve cells there that gives out the command. Just after your central sulcus is your post-central gyrus, and that is important because it is the primary sensory. Okay, that is why your central sulcus is very important um, landmark, because it divides your primary motor cortex in your frontal lobe and your post-central primary sensory cortex in your parietal lobe. Basically, when you talk about your frontal lobe, you always think about motor functions. Well, generally speaking, not always, but anyway, when you talk about your parietal function, you're always thinking about sensory. And that even goes with speech, okay? Because somewhere here is your sensory speech area. What does that mean? Sensory speech area is also known as your vernix area. What does that mean? If I show you something or I show you a word, your speech area basically interprets that word. Okay? In your frontal cortex, you have your motor speech. And what is that? When you want to speak, that's your Broca's. If you want to speak, that's the area that commands your or what words that you're going to say. Okay? If you want to write something, that is also the one that's responsible in forming the words that you are going to write. Okay? And of course, in your lateral portion, you have your auditory here, cortex, and then you have your memory here. Okay, so your auditory cortex is somewhere between somewhere in the periphery of your parietal. Again, it's sensory. Okay, now why am I drawing all this? Because let's see how your brain interacts with the different areas. Okay, so for example, I'm going to do something. I'm going to write a command. Okay. So when you see this command, what do? When you see this, basically your eyeballs process or get the light, receive the light from this one, goes to your retina, goes to your optic nerve. Basically, it goes to your occipital lobe, your primary visual cortex. And once it sees the images, it brings it here to your sensory speech, your vernix area. Why? Because it is language. And it is a language that you can understand because it is English. So, once you process it in your vernix area, you now understand the sentence. And it says, say, ah. Now, once you understand that sentence, you say, oh, this is a command telling me to say, ah. And it brings it here to your frontal lobe, where your prefrontal cortex basically decides on whether you're going to obey this command or not. Sometimes it sends some impulses to your memory, because you might have a memory of a teacher smacking you for not obeying a command. So, okay, brings back to your prefrontal, your judgment areas, and then you're going to say, okay, I'm going to follow. So, I'm going now to say, ah, and then it's going to go to your Broca's. Your Broca's will now formulate basically the commands of saying ah, because it is motor speech. And then it's going to your motor cortex to tell your actual motor neurons there that basically will control the muscles of your tongue, your lips, your jaw, and whatever else. Okay? So this is a very simplified view of your sorry, simplified version of how you're going to your different parts of your brain areas interact with one another. Okay? And remember, every time you do this, every time you engage all these areas in a learning process, you form more connections and you have better memory. Your learning is better. You have more connections. You have more, what's the term there? Um, anyway, I forgot the term. Let's take another example. Let's say I'm going to write a command, or I'm going to say, I'm going to speak. I'm going to give a command in spoken language. I'm going to say, draw a circle. Okay? Draw a circle. So what does your brain do? Now, think with me. Let's make this into an exercise. Once you hear the words, draw a circle, what happens? So, basically, let's use a different color now. Once you hear that, okay, you hear that word, those words, you bring it to your auditory cortex. Your auditory cortex now brings it to your vernix area. It's spoken language, which you understand. And then it brings it here, judgment. Okay. Okay, I'm going to obey the command. And then it's going to go to your motor cortex. And then it's going to command, well, strictly speaking, still going to your spinal cord. And then from your spinal cord, it, forgive my drawing there, but it's going to innervate the muscles of your hand to write or to draw a circle. So that's how it basically works. So we've spent 10 minutes just to show you how your brain basically interacts with one another. So the more you do these activities, so once you're listening, once you're writing down notes, you are forming connections between these different areas. You're forming more associations. That's the word I was looking for a while ago. More associations. You will have better memory of your learning, and you have a better understanding of your learning. So that's why I'm telling you guys, when you're talking about neuroanatomy, it's really good that you can draw. It will help you figure things out better, and it will even help you understand and explain things to others better. Now let's go to our actual discussion on nerve physiology. Let's draw the nerve membrane. Now let's say this is the nerve or the membrane of your nerve cell. So this is the inside of your nerve, and this is the extracellular space. Now, basically, what you have to remember is it is more negative inside the nerve, and outside the nerve, it's a bit more positive. It's not absolutely positive, but it is less negative. The reason for that is the nerve has a lot more proteins inside, and proteins are negatively charged. You have ions like chlorine, okay, and you have, although you may have some bit of sodium inside, you have a lot more sodium outside. And sodium, as we know, is positively charged. There, inside the cell, you might have more potassium than outside, but this is quite small. The big mover of positivity would be your sodium. So remember, nerve more negative inside its cell. It's because of your proteins, negatively charged ions, and the fact that you have so much fewer sodium inside than outside. Now, go back to your um, nerve membrane. You have channels where ions and proteins and whatever substances could basically come in. So, for the sake of our discussion, let's just include the channels that are needed. So, you have your voltage gated calcium channels. You also have your voltage or your um, potassium channels. Okay, and then you have your sodium potassium pump. Okay, so your sodium potassium pump is an active transport. It needs energy, and what it does is it brings back it brings back potassium and puts out sodium. Okay, so it basically brings out or spits out 3 sodium ions and spits in 2 potassium ions. So just by looking at the math, you have your sodium potassium pump is basically returning the homeostasis where there's a lot less sodium and more potassium inside compared to outside. Now, so let's basically this is where all stands. Your resting membrane potential. Okay, so this is your resting potential. The difference between the inside and outside is usually negative 70 millivolts. Of course, well, if you look at lectures online, textbooks, professors in universities, they would usually say it's negative 70. Your Guyton, during my time, also says negative 70, but your newer Guyton says it's negative 90. But to avoid confusion, let's stick with a negative 70. Now, you see this red line? This red line here is your firing threshold or firing level. Threshold. Once the charge of your membrane reaches this, it would now cause a depolarization, meaning your millions of sodium channels will now open and billions of sodium ions will come inside the nerve cell, and your charge inside the nerve cell will be from negative to become almost or slightly positive, and that cannot be stopped. This is this firing threshold is when you reach that all or none. Okay, once you reach this, it will fire. Your nerve membrane will become excited. If not, then it will just get tickled. Now, let's say a small degree or a small level impulse reaches your membrane. What happens? If it's only a very small impulse, very low degree impulse, it will open a few sodium channels and sodium will come in, and your resting membrane potential will probably be a bit more positive. Okay, from negative 70, maybe it will become negative 68 or something. But if it does not reach the firing threshold, what your nerve will do is it will put out potassium, so it would balance it out, and your sodium potassium pump will pump out whatever sodium is there, excess sodium, and bring back potassium. So basically, your membrane potential will return to your resting membrane potential, which is negative 70. Okay, so let's take that away. So what happens when a larger impulse comes in? Let's say large enough that it opened a sufficient number of sodium channels such that your resting membrane potential became less negative and it actually reaches your firing level. What happens next? If it reaches, your firing level is around negative 55 or negative 50 millivolts. Once it reaches that, what happens? It now reaches, it's all or none. It reached the firing threshold now. So there's going to be that all response, that depolarization. So what happens? All your or most of your sodium channels will open up. So sodium will flood in. So once your sodium floods in, your negativity will become a little bit positive. So what happens? You now have a spike. So it becomes less negative and it becomes even positive. This is because your sodium goes inside. Once it goes inside, your nerve cell, in response, it wants to maintain the resting membrane potential. So what does it do? It releases potassium. It tries to open up potassium channels just to counteract your sodium. So your your potassium channels now open at around this part, and because of that, your action potential doesn't go straight up all the time. It slows down and it peaks, and it now goes back down. It goes back down because now you have your majority of your potassium channels getting your potassium ions out, and then your sodium channels now close. So these now close, your sodium stop coming in. Okay, so your cell tries to return to its negative state. So what happens is it becomes a bit more negative until, well, you have some bit of hyperpolarization here. This is the part where the sodium channels also close. But of course, just like a speeding car, you just cannot brake that fast. You have some bit of hyperpolarization here, and then eventually resting membrane potential. So what makes your depolarization occur? It is your sodium coming in. What does this curve what causes this curve? It's because of your potassium going out and your sodium potassium pump. A bit slow. This is a bit slow, but at least it counteracts your spike and it returns your nerve membrane to its resting potential. So basically, that's your depolarization. From resting membrane, you have impulse, and then you have your firing level. Once that is reached, you have depolarization, and then your potassium opens up, sodium channels close. So now you have return to normal, hyperpolarization, and back to normal. So this is basically where your nerve becomes excitable. So just to reiterate, it is your sodium that causes this depolarization, and the repolarization is your potassium and your potassium channels spitting out potassium and your sodium potassium pump being biased towards sodium. Okay, next. Now let's draw your typical nerve cell. Okay, now once this nerve cell depolarizes due to an impulse, okay, what happens is this part depolarizes, and then the nearby membrane eventually also depolarizes, and then what's beside it, and then another. So what happens is impulses travel, or depolarization travels along the axon, because depolarization in one part causes depolarization in another part of the membrane. Okay, there, until it reaches your terminal knob, and your terminal knob probably ends up on another cell, nerve cell, and this one causes depolarization of this, and so your impulse is thus propagated. Now, imagine this method of propagation as a bucket brigade. Okay, one person handing out a bucket to another person, and then he'll give this to this person, he'll give this to this person. Okay, so you have a whole lot of people waiting for that bucket to arrive, and then the last person now empties the bucket. Okay, it's a, well, it's an okay, it's a rudimentary transmission of your bucket, but the problem with that is it would take some time. Okay, it would take some time. So we now have this substance called myelin. Myelin is a fatty insulator. So what does it do? Let's draw another axon. Okay, so basically what myelin does is it coats certain areas, or particular areas of your axon. Okay, so basically your myelin is formed by your Schwann cell. So basically this one is one Schwann cell after the other. Basically, if you look at that, this is your nerve fiber. Your Schwann cell initially envelopes there, and this is the nucleus. Basically, the Schwann cell envelopes a portion of your axon. So what's the use of your myelin? Your myelin, basically, when this part of the membrane depolarizes, this area is affected. So basically, once this area depolarizes, it causes this area to depolarize. Okay, so it basically, this area is affected, and then this again depolarizes, then this depolarizes. So what happens is, basically, instead of having a bucket brigade, you basically have one person tossing the ball, the other one catches it, and then he tosses it again to another one, tosses it, toss. So basically, you have a much faster transmission. Okay, so usually you have this one is around 0.25 meters per second. With this one, depending on the size of the nerve, it could be as fast as 100 meters per second. So just imagine the advantage of speed. Okay, so just for us to remember, recall, these areas, these open areas, they are called nodes of Ranvier. These are carefully spaced out areas such that when one node depolarizes, the area, I mean this node, can affect this adjacent node. Because sometimes the problem is if your myelin becomes too long and your node of Ranvier is too far apart, if your area depolarizes here, sometimes this area is too far apart to be depolarized in turn. So your transmission stops. So your nodes of Ranvier are actually precisely spaced out so that your transmission will continue, but then it jumps from one node of Ranvier to another. And this mode of transmission is called saltatory transmission. Saltatory. And this is much, much faster. So the advantage of your myelin is that it makes your transmission faster, okay, and it becomes more efficient. Why? Because look at this. In this illustration, if you have no myelin, look at how much energy do you need to cause depolarization in all these areas. Because it takes energy for your sodium-potassium pump to open up, for your potassium pump to open up. But in this case, just you have fewer depolarizations going on. So you have less energy used. Okay? So your myelin is pretty helpful because it makes your transmissions better by making it faster and more energy efficient. Now let's go to your terminal knob. Eventually, your axons will, wait, let me make that bigger. Eventually, your axons will end in your terminal knobs, and then most probably it will end up, or it will touch another neuron, okay, just to propagate that nerve impulse. So your terminal knob, okay, this is your terminal knob. Let's say this is another nerve cell, and this gap here is called your synapse or synaptic cleft. Okay, so as we learned before, you have receptors, okay, just to, because this is how they interact with one another. We're going to discuss that. So these are your sodium voltage gated sodium channels, and then in your terminal knobs you have vesicles containing neurotransmitters, substances that are called neurotransmitters. Now, you can see that there's a whole gap in between. Okay, there's a gap here. And the only way for that gap to be bridged is by the terminal knob spitting out these neurotransmitters to the other side. Now, so what happens when, let's say, your impulses reach the end? Once your depolarization impulses reach the terminal knobs, you have special gates here. What happens is calcium channels open. So you now have calcium here. Because you have calcium channels that are now open, what happens is that your vesicles will migrate to the end. Okay, so these vesicles will migrate here, and eventually they will release your neurotransmitters. And these neurotransmitters will head out. It will cross your synaptic cleft, and some of these neurotransmitters will eventually pair up with these receptors on the other side. Okay, so once it does that, once it pairs to the other side, what happens? These voltage gated, basically it will cause another depolarization on the other side. Because of that, your sodium channels will open, and you will have another depolarization on the other side. So that's how your nerves communicate with one another. This one, basically, this is an example of one nerve cell stimulating another nerve cell. Now, the question is it all stimulation? Does your nerve cell always stimulate another nerve cell? The answer is no. And actually, most of your nerve cells don't actually stimulate other nerve cells. The thing is, most nerve cells inhibit other nerve cells. Only a few facilitate, most inhibit. So the higher control that you have, the more inhibition happens. So what happens in inhibition? We've talked about one nerve cell exciting the other. Now we're going to talk about one nerve cell inhibiting another. So it all boils down to what neurotransmitter do you have? The most common facilitatory neurotransmitter is called your acetylcholine. Okay? Now we're going to talk about an example of your inhibitory neurotransmitter. Now we'll draw it differently. It's one example is your GABA. Okay? Gamma aminobutyric acid. Don't worry, just remember GABA. So let's say this is your nerve cell wants to inhibit another. So what happens? Again, there's progression of your membrane depolarization. Once it reaches your terminal knob, calcium comes in. Your vesicles will migrate to the tip. Your vesicles will spit out your neurotransmitter. This time it's GABA. And your GABA neurotransmitters will eventually pair up with your receptors. Of course, we're talking about GABA receptors. And what happens when GABA neurotransmitters pair up with their receptors? your sodium channels do not open. Instead, you have another channel, your chlorine. So, chlorine comes in. And what is chlorine? It is negatively charged. So, if it's negatively charged, your nerve, that inside your nerve is already negative. Once you put in more negative ions, it's going to become more negative. So, you're not going to make this nerve excited. You're going to inhibit it. So that's how it goes. There are two ways of nerve cells talking to one another. One is it excites the other nerve. The other one inhibits the other nerve. And surprise, surprise, most of your nerves inhibit one another. That's what happens when, let's say, you imbibe too much alcohol. Before, you're usually very quiet, you do not sing, you do not dance. But once you have alcohol in your brain cells that usually inhibit becomes inhibited or gets knocked out, and all of a sudden you now feel the sudden urge to sing and dance in the place where you imbibe alcohol. So that's basically it. Just remember the ions that come in, the ions that cause depolarization, and the ions that are used for the cell to return to its normal resting membrane potential. And then also remember this: ion or nerve cells talk to one another. They can facilitate or excite, or they can inhibit. And most of the time, it's usually inhibition. And we'll see that in the future lectures. So that's it for now. I do hope you will learn something, and by drawing things again, you now understand why you understand or learn more when you draw or write or talk about it. So, thank you for listening. Until next time.